She obtained her PhD at the University of Cambridge, and currently she is a professor at the Chemical Materials and Biological Engineering at the University of Sheffield. And today her talk is about the natural polymers of bacterial origin and their biomedical applications. It's a very pleasure to have Professor Roy here. Professor Roy, now please go ahead.
Thank you so much. And it's my pleasure to deliver this talk today. So, as mentioned, my talk is about the natural polymers of bacterial origin and their biomedical applications. So, gosh, it's not moving. Let me see. Yeah. So there are 4 polymers in my group at the moment that I work with: polyhydroxyalkanoates, bacterial cellulose, and gamma polyglutamic acid and alginate. All of these I produce using bacterial fermentation. Polyhydroxyalkanoates, or in short PHAs, have been my focus for about 25 years now. Bacterial cellulose, I have about 10 years of experience working with it. And gamma PGA, or gamma polyglutamic acid, and alginate are newer members of my group. So the general structure of polyhydroxyalkanoates, it is basically a polyester, as you can see. We have a variety here in the sense that the side chains, that is R1, R2, and X, can vary. And therefore, what we really have is a family of polymers. The short chain length PHAs, as we call them, have monomer units going from C4 to C5, whereas the medium chain length PHAs, as we call them, are mainly monomer units going from C6 to C16. And normally the short chain ones are hard and brittle, although there is a P4HB, which is FDA approved for medical applications, which is highly elastomeric. And medium chain length PHAs are always highly elastomeric in nature. We produce these polymers in large scale in Sheffield. We have a 30-liter fermenter as a pilot plant. And when I first started working on these polymers for medical applications, I chose Bacillus as the main organism for production. It's a gram-positive bacteria. It doesn't have lipopolysaccharides in its membrane, and therefore it would be non-immunogenic in nature. I'm sorry, there's a background sound coming from somewhere. My apologies. Yeah, okay, that's good. So in the gram-positive bacteria, we do not have lipopolysaccharides, which are known immunogens, and therefore the polymer that you produce would have no risk of having any lipopolysaccharides, and therefore would be excellent for medical applications. So this was my reason for focusing on Bacillus species, and I mainly produce short chain length PHAs using this species. So this is a typical temporal profile of the polymer production. You can see that with time, the green bars are increasing. That is, the polymer yield is increasing. The bacterial cells are growing, as you can see with the orange profile, and overall is a highly controlled process. And this is one main reason I use fermentation for the production of natural polymers rather than isolating them from a natural source, because I have exactly controlled conditions, and therefore the polymer that is produced has the same molecular composition, molecular weight, and this is essential, as you might appreciate, for any biomedical application. So after I produce, I always make sure we have the right structure, and this is a C13 and a proton enema of the polymer, and a GPC, which tells us the molecular weight of the polymer, because these are so crucial for the polymer to be used in biomedical applications. So this is a typical example of P3HB that I produce in my group, and you can see that it is a high melting and a very stiff polymer. So the Young's modulus is about 1,000 megapascal or a gigapascal. However, for MCl PHA production, I have to use gram-negative bacteria because gram-positive bacteria do not produce MCl PHAs, and the main species I have used is the Pseudomonas species. I've produced many different types of medium chain length PHAs and again used controlled fermentation for the production. So you can see here there is the FTIR spectrum of the polymer. Here is the NMR, and in this case we also did a 2D NMR to ensure that the polymer that we produce is absolutely the same as we desire to produce. Here is a GPC, and you can see that the molecular weight is also measured every time we produce the polymer. This is a typical medium chain length PHA, and here you can see is much lower in melting point, only about 53, and the Young's modulus is only about 15 megapascal, which is again much lower than P3HB. So although both of these polymers are polyhydroxyalkanoates, you can see how different they are as far as the polymer is concerned. I've also started using the sunshine in England. Well, we don't have a lot, but whatever we have for algal production of PHAs, and this is in collaboration with Professor Jags Pandal. So what do I do? Once I get the polymer, the next thing is to make things out of it. And for that, we need to process the polymer. Many groups, including ours, we have now processed PHAs using all the traditional manufacturing methods and also the upcoming new methods like SLS printing, FDM printing, etc. So these are the processing methods mainly that I have used in my group. So we have PHB porous structures, we have microspheres for drug delivery, we've made gyrospun fibers in collaboration with Professor Mohan Edarisinghe. We have 3D printed structures, we have tubular structures, we have melt electrospun sheets, we have stents, we produce stents by melt extrusion and laser ablation, and 3D molded structures. So there are many different processing methods that we have used, and here you can see the FDM printing of medium chain length PHA, and you can see how beautifully elastomeric and it's so malleable, and it is also very suturable once it's 3D printed. So this is 3D printing of MCl PHA. This is yet another MCl PHA, not the same one as you saw before, that we have managed to 3D print, and you can see that structures are very, very clearly defined and robust. This is P3HB. We have managed to print P3HB really well. There is always a kind of belief that P3HB cannot be printed very well because it degrades very close to its degradation, to its melting temperature. But we have used certain specific methods in order to print them successfully. The bottom ones are composites with titanium oxide that we created with P3HB. So the main message I have is PHAs are the new emerging medical materials. As some of you might know, this has been cleared by FDA for medical purposes, and TIFA is a company based in Boston that have mainly worked towards this. And hopefully, I will tell you later about my spin-out company, and we shall take further the PHAs into the medical applications. So I'll give you a few examples. I've been given 20 minutes, so I have about 15 more minutes to tell you all about what I have done. So I'll briefly touch upon a few topics. Bone tissue engineering. The first type of application that I explored was bone tissue engineering. And here is one example. This is gyrospun scaffolds that were produced with P3HB and hydroxyapatite. The fibers were produced, as you can see here, with a rotating cylinder, and the fibers are gyrated out of the cylinder. And you can see how beautifully beautifully aligned these fibers are, nice and porous in nature. And when we did some egg studies, which is an ex ovo study, these are called the CAM studies, to determine the biocompatibility and the vascularization of these gyrospun scaffolds, we obtained beautiful results. This is a research we did in collaboration with Professor Richard Orefo, and what you can see here is that there is beautiful vascularization of the prototype of the scaffold within this environment. And when we did the staining with H&E staining and ANS staining, we saw excellent vascularization. The red indicates the blood vessels and the cells, and the blue indicated the collagen formation. So this told us that this is a wonderful material for bone regeneration. We then went in vivo in rat, and this was then seen that the top particular row here is P3HB only, gyrospun fibers. This is P3HB with STRO1 cells, which are bone cells, and you can see that the cells are growing, attaching really well, with STRO1 cells already seeded onto the P3HB. Then the next one is P3HB and hydroxyapatite. You can see here that the vascularization is a bit more. And then finally, we did P3HB-HA with the STRO1 cells. And you can observe here that we got beautiful vascularization and collagen formation, indicating that these gyrospun scaffolds are excellent for bone regeneration. We've also done recently some work with inkjet carbon black additive, which we added to poly-3-hydroxybutyrate. This was work done by Dr. Andrea Mele from my lab. And you can see that he managed to 3D print them beautifully with high fidelity. And in the presence of the inkjet carbon black additive, we got high porosity, as you can see from the micro-CT image here, and also the fidelity of printing enhanced. So this was also found to be very, very biocompatible. You can see primary osteoblasts which attached and grew on these 3D printed structures, and they even in fact jumped the gaps that were, or the pores that were present in the scaffold, indicating that they really liked the whole substrate, and again showing that these polymers are excellent for bone tissue re-engineering or engineering. Here we have further evidence. You can see that mineralization has occurred, which is important for bone. As we know, it's collagen plus hydroxyapatite. And this was a xylanol orange assay that we did and found that we got excellent mineralization in both P3HB and the inkjet composite. However, the inkjet composite was a bit better. TCP, we got nothing. And when we did the alkaline phosphatase assay, again, the same results were obtained, that P3HB showed ALP activity, and the inkjet composite also showed activity, but even higher. And ALP, or alkaline phosphatase, indicates the mineralization or the formation of calcium phosphate within the tissue. So these results told us that these polymers are excellent for bone tissue engineering. I'll now move on to peripheral nerve tissue engineering, and this work that I present is in collaboration with the University of Torino in Italy. And here you can see that when nerves break due to trauma or accident, if they are anything more than 5 millimeter, they don't really repair very easily. So the idea is to create a bridge, to suture a nerve conduit on top of the nerves, and then allow the nerves to regrow and regenerate. So we produce these tubular structures with medium chain length PHA and P3HB blend, and you can see here how beautifully they caused a regeneration of the nerve within 6 weeks. So the suture, you can see, attached the nerve conduit, and the nerve then grew back within 6 weeks. And here is the nerve after 12 weeks. They are very well regenerated, and with the conduit, we got the regeneration, as you see on top, the nerve guidance conduit, which is that tubular structure that I mentioned to you. And this is with an autograft where the nerve itself from another region of the animal was placed within the broken nerve. So you can see between the conduit and the autograft, we got almost very similar regeneration. And here is the proof, that is, the rat could then grasp and really dance, as you can see here. And this sort of functional recovery was observed with the nerve guidance conduit within 9 weeks, whereas the autograft and comparable to the autograft. And I think this is a fabulous result. And here we did also another study with a European project in collaboration with many partners. And here you can see we took a blend of P3HO Co3HD, which is the hydroxyoctanoate decanoate copolymer, a medium chain length PHA, blended it with PCl, and those are the tubes that we presented or we put within the broken nerves. And here we had an autograft, a silicone tube, and Neurolac, which is the currently used nerve guidance conduit by the surgeons. So when we did the immunohistochemistry, we observed beautiful regeneration in all the cases. So we had different types of tubes, those that were 500 micron in diameter, those that were 250 micron in diameter, with and without pores. And in all of them, we got really good nerve regeneration. And finally, when we compared them across all the types of samples that we observed, we found that the blend of P3HO-HD with PCl was almost the best, except for the autograft, which obviously is a real nerve, and one can't expect that we are absolutely the same as an autograft. There was no significant difference, but we could see that it was much better than Neurolac, which is the current commercially available nerve guidance conduit to the surgeons. Cardiac tissue engineering is the next topic. Here I collaborated with Imperial College London, and here we had a regenerative medicine center, and we observed or we tried to create what is called a cardiac patch. Post heart attack or myocardial infarction, what happens is that the heart, the left ventricle side of the heart, as you can see in this diagram, develops a scar tissue. And therefore, the heart is not able to pump blood very efficiently. As you can imagine, a scar tissue is very tough and stiff. It doesn't allow the heart to pulsate and pump blood very well. And therefore, what happens is the heart starts becoming bigger in size, and finally it's a failure, the heart failure, and the patient dies. So our idea was, can we create a patch which contains cardiomyocytes and patch the heart, or put this patch onto the scar region such that these cells would transfer to the heart region, and we would then end up getting healthy cardiomyocytes replacing the scar tissue, and therefore making the heart again functional as before. So look at this here. We've got iPSC derived cardiomyocytes, which are very nice stem cell derived cardiomyocytes, and here is an adult cardiomyocyte. You can see how different they are. So what we tried to do is we used the iPSC-CMs, but took medium chain length PHA that we had melt electrowritten. And after melt electrowriting, these are the structures we obtained, which means we had long and thin struts on which the cardiomyocytes could sit and elongate and therefore mature like a normal adult cardiomyocyte. And that's exactly what we saw. So you can see here that there was a beautiful anisotropic development, and these were highly mature cardiomyocytes that we found. And when we looked at calcium transients, which is the main functional assessment of cardiomyocyte functions, and observed that we had excellent, normal calcium transients, we, of course, understand that any patch needs vascularization, which is a major challenge for tissue engineering. So what we did was we tried to grow iPSC, again, the induced pluripotent stem cell derived endothelial cells, and observed whether we could get some nice, vascularization. And you can see here that the endothelial cells grew on the medium chain length PHA and also resulted in vessel formation. We then combined the two types of cells and we created the patch. This is an acellular patch that we patched onto a rat heart, and you can see here that we had excellent acceptance of the patch. There was no immune response, there was no capsule formation. Then we put the cell containing the induced pluripotent stem cells and the endothelial cell containing patches onto the heart, and this in vivo work is continuing now to see whether this will lead to functional improvement. The other, I only have 2 minutes left, so I'll quickly pass through what else I have done, a few of the things like kidney tissue engineering. We're looking to create a filtration barrier within the kidney, and here we have the polymer over which we have grown these kidney cells, the podocytes, the endothelial cells, co-cultured them, and we have observed excellent growth of these cells on our 3D printed structures. So here you have a 90 degrees strut, we have 60 degrees angled strut and 30 degrees, and all of them we saw very nice collagen formation and cell growth. We are also trying to develop bioartificial pancreas. Here again, medium chain length PHA has been used, and we have observed, and what we did was we seeded Brin BD11 cell lines. We co-printed them along with alginate, encapsulated in alginate, and we observed that these cells were producing insulin in a glucose responsive manner. And these were the cells on the constructs. As you can see, they're founding or forming really nice agglomerates, which is typical of pancreatic cells. We've also done some experimentation where we've added calcium peroxide to these scaffolds in order to provide extra oxygen to these pancreatic cells because they need a lot of oxygen. We've done controlled drug delivery, we've made stents like I mentioned earlier, and just 2 slides on bacterial cellulose that we produce again using glucon acetobacter xylanus, and here we have beautiful microfibrillated bacterial cellulose. We have tried to scale up this production and have produced antibacterial bacterial cellulose. As you can see here, the surface of non-functionalized bacterial cellulose and with the Staph aureus growing on it very happily, whereas when we functionalize them, as soon as they attach, they grow, they cannot grow and they lyse. We also now making wound healing patches with these bacterial cellulose and alginate blend. And finally, I'd like to conclude my talk by saying, I hope I've convinced you that PHAs are an emerging class of sustainable, biodegradable and biocompatible polymers of natural origin with huge potential in biomedical applications. We have scaled this up now to 750 liters, not just 100. We have used PHAs for drug delivery, hard and soft tissue engineering, wound healing, bioartificial pancreas, stents, nerve conduits, antimicrobial polymers, and even 3D models for cancer. Bacterial cellulose is another natural polymer that is emerging in my laboratory, and here I've shown you how I've used it for wound healing. I'd like to thank all the key scientists and collaborators for this work, the funding bodies, of course, my research group. And I'd like to also tell you that now we have spun out a company called FAST from the University of Sheffield, where we are now producing biomedical grade PHAs and for use in biomedical applications. And thank you for your attention.